Hi everyone, I am Octavio Zambada. Currently, I am studying a master's degree in plant biotechnology at PSTVd-tomato. Today, I am presenting a study centered on BHLH transcription factors and the regulation mechanism, as well as their role on pathogenesis in the bipartite tomato model, and how this mechanism can be different depending on tissue and infection type. In previous studies, we have identified the relevant participation of this transcription factor family in orchestrating viral disease as master regulators. In order to study unique and conserved features of regulation, we built a gene co-expression network that contains all the transcription factors of tomato known to date by using public time-course microarrays datasets for two different tissues, roots and leaves. The interactions for BHLH transcription factors inferred in the gene co-expression network as annotation files, sample phenotypes, and the gene expression matrix from microarray were used as input for SemiTool, a systems biology method that easily identifies and performs comprehensive modular analysis. As results, we obtain a gene set enrichment analysis in which we can observe the behavior of these models' gene expression through the infection stages, gene ontologies for each model, hop genes, which are defined as genes with high connectivity and importance in the model, and a visualization of the network for each co-expression model in the dataset highlighting hop genes by their interaction and co-expression rates. Using these last two outputs, we proceed to find more features for the models. First, we identified microproteins, which are a group of proteins with transcriptional regulatory potential. Then, we built a bipartite network of the most interesting models. These are the models that showed a very differential expression rate among infection stages for each tissue. This allowed us to have a better understanding of the regulation mechanism implied inside them. Finally, we looked for the presence of the bifan motif in the model's gene co-expression networks. This is a very specific structure in graphs that explain a certain type of regulation when we are talking about transcription factors and their targets. Overall, we found that BHLH transcription factors are regulating distinctive activities depending on the studied tissue. In roots, they have a regulatory activity in water and ion transport, which could be a mechanism for fast lipid membrane repair in root tips. And on the other hand, in leaves, they are implied in the formation of wax to protect them. It is also observable that the regulation of some specific pathways seems to have a very complex regulation, with up to 4 different BHLH transcription factors taking part of it. For instance, this behavior is appreciable in roots for photosynthetic processes and in leaves for cutting, suberin, and wax biosynthesis. Thanks for your attention and have a nice day.